questions are coming, but I have a number of questions actually to the different. So the first question I have is for Terry. In view that we have this new information about the um, polycystin 2 channel activity, that this is not a specific calcium specific channel. Like we had, because in the past, there's been a lot of thought about calcium channel blocker as a class as not preferable for ADPKD. Would this new knowledge change the way you think about the use of this class of agents? As for treatment of hypertension.
For hypertension.
Yes.
Well, I would say that most of the data against calcium channel blockers was like pouring stuff on cells and culture. And I don't know, even forgetting about the new data, that replicates the situation in humans to begin with. And then going to the data that I presented, as I understand it, I don't think it means that polycystin 2 is not a calcium channel in the cell. I just don't think we know yet.
Mm-hmm.
We haven't figured out in the in vivo situation what exactly is going on, right? Because some of the, well, as I understand it, if you were to be able to get a high calcium concentration in the cilium, that would activate the channel and increase calcium influx, but we just don't know if that is exactly what's happening. So for me, I have two other experienced clinicians. If I need to use a calcium channel blocker, I do.
Any comments from Ron or Vincente?
Well, I generally avoid using it as first-line therapy, but it's also important to control the blood pressure. And the hydropyridine calcium channel blockers were used in HALT as, I think, third or fourth-line therapy. So again, you've got to control the blood pressure, but I would start with ACE or ARB.
I do too, but...
No, I agree. I start with ACE inhibitor or ARB, and I like drugs that combine alpha beta blockers like carvedilol, so that's frequently my second choice, but I have no problem using calcium channel blocker if needed or if there is a different indication for it. And I think the dose that we use is not comparable to the concentration that has been used in vitro or the doses that have been used in vivo in animal models. So I don't have any problem with it.
Actually, I have a second question for myself. So actually, it was one of the comments I find. Oh, sorry, great. Do you want to say something? So I wonder, as we understand the genetics better, maybe we will start to revise our thought about so-called simple cysts. Maybe they are not so simple. They may have a hypomorphic PKD1 mutation, because we do see a number of individuals that we would say these are simple cysts, but they do carry a missense PKD1 mutation that would be predicted to be pathogenic and seem to be very mild. So it adds as a potential modifier, but it really doesn't cause disease. But if this is frequent enough, it can cause like a variability in people who have this kind of diagenic inheritance on a background, more conventional PKD mutation. Are you ready, Winnie? Okay, I have more questions. Okay. So for so the about okay. So this is a question for Tim. So for the automated algorithm, I guess when you look at follow-up scans, you want to align the first to the other.
Yep.
So one question is that because some patient now we're doing sclerotherapy. Have you had any experience once you have done sclerotherapy, you take out one cyst and the tissue start to change? Can you actually align the previous one to the next one, like to try to make sense of what's happened there?
Yeah, so we have done a little bit of that. I would say it's more been visual, so we can, essentially align the scans and get a sense for what the changes are. But I think there could be as you're hinting at, some interesting ways that we could do this kind of change tracking and quantify those changes, what's happening in the local environment when you're going through those procedures. But largely it's been more kind of, before, after. We do things like measure the cysts within the kidneys, but nothing kind of quantified in that sense. But I think that would be a very interesting thing to do, trying to look at even individual cysts, how those are growing or changing, and figuring out ways that we can visualize that.
Right.
It's been a challenge, but it's something that we're certainly working on.
Okay, Ron.
If you don't mind, I have another question for Tim. How do I get automated segmentation into my institution? Because we tried working with you a long time ago, and our IT folks were very timid about introducing, active software into our system.
So I can comment that that's a question I often get as well. And I would say that, we've run up against a number of different hurdles depending on the institution. I'm more than enthusiastic to try to get it, working at each of the institutions. Like, for example, I have an extended stay here in Toronto to work a bit more with what we got set up here. But, it probably would be something that we should have a meeting with the IT folks again and see, because we've run up against legal issues, and et cetera. And we're not a company, right? We're trying to just get this in your hands so you can use it, but would certainly be open to those discussions with anybody interested. I apologize that it's been a conversation ongoing for a while, and we would like to find a solution. So yeah, I appreciate it.
And then can you put it into the cloud? And so work in the cloud environment where you can upload your information. I mean, processing wise, be a heck of a lot faster, right?
Yep. Yeah, I can comment on that. Actually, that was one of the first directions we tried to go, but that's where we ran into a lot of the legal issues with institutions being concerned about, obviously, sending images out that could potentially have PHI. So one thing we had initially looked at is, helping with that kind of de-identification process, and then running the software. Because actually a lot of the tools, I think, that are coming out now, that's what they're doing as a solution, because then you can have a stable kind of build of the software and run it that way. I think that's somewhere we would like to go, but with the initial discussions, it was just such a battle from the IT side at the external sites to do something like that.
It wouldn't be like you couldn't have like a platform where they could have their own unique, where they would have their own little space, so they would be not sharing their data, but you'd be able to put the software on top and do the analysis.
Yeah, I mean, that is definitely something we want to do. I think it's a question of how do we best make a solution that works at as many institutions as possible. And that's what drove us to this particular solution of just bringing the tool in-house and then trying to get it set up, with the specific images at the site, et cetera. But yeah, I think that's somewhere we'd like to go. It was just that was actually the initial direction we tried to go, and we ran into a wall. So unfortunately, yeah.
So we have some question from the audience. We can go back and forth. So the first question is, does getting a genetic test to determine someone's specific genetics, PKD 1 or 2, have any benefits from prognosis or disease management? So as I mentioned, I think we could predict kind of ballpark effect, like the group of patients that have certain mutation class, like whether they would likely do well or not. But for individual patients, because of the very significant modifier effect, it is not completely reliable. And this could probably also related to this pro-PKD risk assessment, which kind of use genetics as well as some of the clinical data, such as early development of high blood pressure or hematuria, and it has been shown to have some predictive value in a single center study and not been validated. One of the problems with the pro-PKD score is that often we don't have reliable information on this clinical risk data. Patient may not remember whether they have early onset hypertension or when they have the first episode of hematuria. So they're a very soft sign, and but it is certainly one of the approaches that had been proposed. I'm going to go back and forth. I make some notes. So I have one question for Vincente about the actually your comment about the reprise study, that there was no significant benefit in those over age 55 years. On the other hand, the sample size for that group is about 100, which is about 1/5 of that bigger group that have. So I wonder whether it could also be a problem with this much smaller sample size and demonstrating benefit is very difficult. Like,
no, I agree. I think the 55-year-old limit is a relative limit, and I think in individual cases, you can have to judge many other elements to decide whether it makes sense or not. But there are a number of issues. One is the sample size. Mm-hmm. The other issue is that the patients that were over 55, on placebo, had a slow rate of progression of the disease. The criteria that they were used to enter the reprise study did not include total kidney volume, were based only on GFR. So it's possible some of the patients that were entered with a low GFR, they had multiple cysts in the kidneys and other causes for the GFR being down. So it was maybe not as clean group as the patients under age 55 that had other criteria they had rapidly progressive disease, obviously. And so I think there are a number of explanations. So I would not say this 55 years old limit is in a stone. It's kind of something that is a consideration. But if I have a patient that is 66 years old, I have a GFR of 27 or 28 and has kidneys that has a mutation in the PKD2 gene or a PKD1 non-truncating mutation, and the kidneys are not that big and has been hypertensive for about 40 years, and the hypertension may not have been very well controlled. I would not probably be very enthusiastic about tolvaptan. My recommendation would be against it. Mm-hmm. And if the patient would push for tolvaptan, I say, well, it's your decision and it cannot be fine. But so it's kind of where you have to use a little bit of clinical judgment and no go just rigidly by age 55. Right. But it's a very difficult question to answer.
Yeah. Yeah. I mean, that's our approach as well. Like after 55, we tend to individualize and discuss because I do also worry because some strong statement had been made that you should not treat those patients. So we have patients that have Mayo Clinic class 1D, who's 56 years old, and the kidney function is half normal. I just feel it very difficult to say, like, not to treat this kind of patient if they have the possibility of accessing the medication. Yeah.
I'm completely in agreement with that. Yeah.
Yeah. Okay. So this may be probably a question for Ron.
Can I make one comment?
Oh, sure. Yeah.
So the age and GFR limits are not in either the U.S. or the Canadian labels. Mm-hmm. Right. So these, I think, reflect the characteristics of the patients in the clinical trials. So right. I agree with Dr. Torres that you can use some judgment, especially in the older individuals.
Right. Yeah. So actually, this question is for you. So any comment on pressure effect on GI tract, especially early satiety, bloating, and heartburn? I presume this is related to organ enlargement, people who have big liver or big kidney.
Yeah, I think that pressure effects are relatively common in people with organomegaly, and we typically advise them to eat smaller meals more frequently and end up prescribing acid blocking medication to them. And depending on the characteristics of the kidneys and or the liver, there might be benefit to either sclerotherapy or other interventions.
Okay, thank you. I have a question. Is it possible to move the questions up? Are there more questions? Okay. Okay, so this is a question for the panel. I think if I read it correctly, and your handwriting is not the best. Okay, so I think I will guess what yours. Okay, so I think is it AI? Okay, not L. Okay, so AI has proven useful in other domains of drug discovery. Is anyone doing that in ADPKD? Do you know of any? I have heard something in the FASAP meeting that some company use an AI assist approach.
There's a company that's using it. Or promoting the use of it. I forget their name though. Maybe they would be in the FASAP program and we could figure that out.
I was going to say that Daryl Quarles at Tennessee recently asked me about that. He apparently is trying to do that and wanted to know if anybody had a target that he could practice or try on. So
It was a thought experiment, really, what they presented.
I think AI is being utilized to design drugs
Right.
for the right receptor for this. I think is being used, but I don't know that is currently specifically used for PKD.
Yeah. So I have a question for Dr. Torres. Actually, Dr. Torres, I have several questions for you, because this is very common from patients that I have seen, and they have asked me that. So because you have shown one of the curves that showed the long-term effect of tolvaptan, like it's an extrapolation using the Tempo 3-4 reprise study, that if you have patients who have normal kidney function to start the drug and continue the drug treatment compared to the control, like assuming that there's a kind of a linear decline, constant decline, that you would have most benefit, like 7 or 8 years. And so the question is, do you have any information on long-term tolvaptan treatment patients, whether their trajectory of kidney function decline is indeed linear? Because in the other studies, for example, the natural history study, I think that maybe Ron, you showed of the JCI Insight paper from the Peter Harris group, like or Alan Yu, like the trajectory seemed to be curvilinear rather than linear. Like, yeah. So then, if it's not linear, how can we be sure that that about this kind of benefit can be sustained?
Well, the patients with Mayo Image Class 1E, the trajectory is almost linear, and D is a little bit less so. But once these patients get after age 30, 35, if you look at C, D, and E, it seems to be quite linear. Maybe not. So the other thing is the information I have is only the study that we did already a few years ago, that I think we look at patients that had been on tolvaptan for up to 11 years, but the average duration of tolvaptan was about 5 years or 5.5 years. And in these patients, we compare them 1-to-2 ratio with participants in HOLT and CRISP, so that's a historical control. And they did better by the expected difference of about 1, slightly above 1 ml per year. But also we did is predict where they should be based on the predictive equation derived from the Mayo Image classification. That's even weaker than historical control, but they were doing much better than it would have been predicted. So that's another way to suggest that is favorable. But the way the question is, do I have a way to prove that these 7 or 8 years, 6 to 7 years of gain of life of kidney function, that this is happening right now? There is no way to prove that. We don't have enough longer follow-up on these patients. So,
Did you want to make some comment, Ron?
No, I agree with
Okay.
what Dr. Torres said about the trajectories.
Right. Okay. So question? Okay, so let me just. So this is a bit of a long question. Oh, sorry. So this is a question. As an ADPKD sufferer, 47-year-old male, PKD1 truncating mutation of various PKD therapy currently under development. I'm most intrigued by the mRNA-based therapy. I think probably you mean the microRNA-based therapy being developed and trialed by Regulus and partner, mainly because it's therapy that go further upstream to the production of polycystin protein. Although it is early and its safety and efficacy have not been demonstrated, I'm interested to get some feedback, interpretation from the presenters, their interpretation of this type of therapeutic approach and other like it, the prospect of them reaching full-scale implementation. Anyone want to try to?
So I think, in principle, it would seem like a appealing therapy because it not only would target PKD1 and PKD2 in terms of upregulating their activity, but what's really quite remarkable is even in the context of a PKD1 model where they just boosted PKD2 activity, they were able to see some benefit. So it was really quite dramatic in the animal models. Let alone it also seems to have an effect on the metabolic pathways by upregulating PPAR alpha activity. So it would seem like it would have a lot of good targets. I think the challenges are one, we're not mice, so we have to see whether it works in humans. Two, I think getting it to the kidney cells, being taken up by kidney cells at a high enough level that it can be effective, I think is part of it. Another challenge will be that it has to be taken regularly. It is going to be presumably an injection of some sort. That you'd have to take it and get it regularly because it only works while it's around. So it's not like a gene therapy where you put it in once, or gene editing, you put it in once and you've got it done. This is something that will be repetitively have to be administered. So there are some challenges yet ahead, but in principle, if the preclinical data is predictive, the antisense oligotherapies are being promoted in other conditions. In SMA, a different genetic condition, for instance, there's been, I think, a therapy recently approved in the last year or two for slowing progression of disease. And so I think that as a class of therapies, there has been evolution in that space. I think it remains to be seen whether all of those properties can work in other models of PKD, other different models of PKD1 and PKD2, just to be sure that it's a robust finding, because it is a very dramatic result.
Ron.
So just a comment. So they've completed a phase 1b study in humans, and they've shown that they can get PC1 and PC2 into exosomes. They then discontinued development of the particular agent they were using. I mean, all this is in press releases. And I believe they've identified another compound where they're, I believe, going to be doing another phase 1b study. So the promise remains undefined at present.
Yeah, I think it's a very exciting approach. It's the first therapy that actually goes to the root of the problem. But it's a phase 2 study, very preliminary. I think I would really encourage patients to participate in this study because I think it may have a great impact. On the other hand, one of the concerns is that this anti-miR-17 may also do other things that we don't know, and that's the safety of it will be something that will have to be evaluated very, very carefully.
One other thing is, the miR-17 levels go up once you have a cystic progression already underway. And that's how then the miR-17 levels go up. And then by blocking that, you then change the, you modify function. The challenge here is, you want it, if you suppress it, then miR-17 is not high, and therefore the drug doesn't work. So it's a little bit, there's some details yet to be, I think, understood in how this really all works, I think it's fair to say.
Yeah, I'd like to just mention that because I get a lot of email from patients almost every month that have promising new therapy. Should I do anything and so I understand that everybody want to be hopeful, and I think we are in an era in which that there are many new approaches that had very promising preclinical study, but translating it to human is the most difficult step, where most of the failure occur, like 80-90% of the promising therapy in the animal model fail when it get to the human. That's why you have to do clinical trial to make sure that the drug actually does work and is safe. And so unfortunately, there's no shortcut. And I think that,
So one of the challenges in this space in terms of translating from preclinical to clinical is that unfortunately, a lot of the preclinical science, has a relatively low reproducibility outcome as one of the challenges. And it gets to, again, failure to accommodate or anticipate the variability of the models that we use in the first place. So the lens, as I said, the size of the studies are often quite small. And we know there's a lot of variability in these animal models. And then I think the other important parameter is just, what is, slowing it in a mouse by 3 months or 6 months or 9 months. We have no idea. We make assumption that it's a relative proportion to your lifespan, but we don't really know that's true at all. And so I think this is again, another reason why therapies may fail, because the benefits may not be quite as dramatic when you make that conversion.
thank you. So let me go to the next question. So could Dr. Torres comment on dosing of tolvaptan? If someone has a urine osmolarity of less than 280, would one up titrate the dose?
Well, depends. One answer would be that in the clinical trial, we increase the dose to tolerability. So we increase the dose all the way up to 30 milligrams daily. And so based on evidence, that should be the goal in clinical practice as well. And if a patient is able to tolerate this dose and without having a major impact on their quality of life or having an impact in the quality of life, then I would suggest that that should be the target. But in patients that cannot tolerate this dose, and I would be satisfied with taking a dose that is sufficient to maintain the urine osmolarity less than 280 before taking the morning dose. For patients that are taking tolvaptan, one thing that is important that's a little bit disconnected from the question, but is a mistake that is frequently done by patients taking tolvaptan is for the safety labs. They take the tolvaptan in the morning and have the blood sample 1 hour later, and that's not a good thing to do. The blood sample and the urine sample have to be taken in the morning before taking the dose of tolvaptan. Otherwise, you have a lot of variability in the results of a blood test, and the urine test, of course, is completely unreliable.
So the next question probably, I think is maybe for Ron or Vincente. So this is a 47-year-old patient from Buffalo. So is regarding the chemistry of kidney stone in ADPKD patient, is there any way to determine, I guess, the composition by non-invasive imaging or study or modality, or is it the only way is to actually analyze the stone pass or remove surgically? So that's one question. And then what kind of procedures are typically used in the event of stone-related complication, such as obstruction, given that all procedures available to healthy individual are not usable. So I'm not sure whether that is indeed true, but maybe I wonder if you can comment.
Well, in regards to the first question, the only way to differentiate the composition of the stones is doing dual energy CT. That's a special CT that you can separate clearly the calcium containing stones from uric acid containing stones. Also, the cystine stones have somewhat different appearance, but the cystine stones are very rare, so usually are not an issue in polycystic kidney disease patients, but certainly you can differentiate calcium from uric acid, and that's an important differentiation because the uric acid stone you can dissolve with medical therapy, whereas the calcium stones, you either leave them alone if they cause no symptoms, or you need some type of lithotripsy or intervention if they give symptoms. So again, I think for a calcium symptomatic stone that is not uric acid, that cannot be dissolved medically, then the only procedure you can do is either lithotripsy or percutaneous lithotomy, or taking the stone percutaneously through a tube, or doing a ureteroscopic and destruction of the stone with laser. And I think any of these procedures can be done with polycystic kidney disease patients. They should be done by a urology group that is expertise with these procedures. And of course, the least invasive when it's possible is the ureteroscopic approach with the lithotomy.
The only thing I would add is that sometimes it's useful to do a 24-hour urine collection to assess for stone risk factors. So if there's hyperuricasuria or elevated oxalate or calcium, then you potentially have interventions. But as Dr. Torres mentioned, only the uric acid stones can be treated completely medically.
One clue that is not definite, but patients that are uric acid stone formers, usually they have a very acid urine. So you have a stone and you have consistently urine pHs of 5.5 or 5.4 or 5.3, then uric acid comes, whereas the urine pH is normally in the above 6. It's not likely to be uric acid stone, but that is not 100%.
So next question is, at this year's ERA conference, doubt was cast on how best to dose tolvaptan, suggesting it would be best to increase to the highest possible dose. Should we strive for highest possible tolvaptan dose tolerated by patient, or any dose that achieve urinary osmolarity target less than 280?
I think I have already answered this question. I think nobody knows whether targeting to maximal dose is better than just targeting to less than 280, because this was never compared. Because in the clinical trial we try to go to maximal dose. In theory, it's better to go to maximal dose if possible, but it's quite likely that you may have similar benefit just aiming for keeping the urine hypotonic completely for 24 hours without really going all the way up. But no one can answer that question, I think, unfortunately. But the safest answer is the clinical trial showed that. Just do it in the clinical trial.
Right, thank you. I think the last question is, any experiments into using mRNA technique to boost PKD1 and PKD2 levels? I guess the microRNA approach is in a way boosting the
But the question is, yes. And I think given all the focus on using mRNA now or RNA based, for instance, we use it for COVID, right? The vaccines are based on RNA therapy. So it gets taken up by the cell, gets made into protein, and you get then your immunization that way. And so the question is, could you do that with with PKD1 or PKD2? PKD1, it's a 14 kb mRNA that is going to be a synthetic challenge. And again, getting it taken up by the cells, presumably liposomes or something, you could put it into an emulsion, maybe you could pick it up, but you'd have to have a constant, again, problem with that is you have to have a constant source of mRNA being delivered to the cells. Assuming you could get the mRNA to the right cells at a high enough level, you'd have to do this, over a very long period of time and regularly, not just like a bolus and then wait for 2 weeks. And so this is, I think, would be a difficult strategy, I think.
Okay, so any other question from the online or the face-to-face audience? Marshall?
A lot of the talks we saw here, comment kept reoccurring that many of these either biomarkers or in a lot of the talks we saw here, many of the comments were that either the biomarkers or the genetic markers were valid across population, but it was much more challenging on individual patients. So, for example, total kidney volume was one of them. So is there anything, or what's your thoughts on what could potentially, or what technique we could potentially use to get a more targeted, patient-specific approach?
I mean, I guess I'll purely from the imaging side. I think that's why we're driving at trying to pull out more information from the individual scans in terms of sort of quantifying those phenotypes of the patients, right? Because obviously total kidney volume, it's really a crude measure of what's going on, visually that clinicians are assessing. I don't know if I have a clear answer to that, but I think trying to drive at more things that we can do, like pulling out information about renal blood flow, pulling out information about fibrosis and scarring, really these underlying mechanisms that we're not really probing, because I see a lot of times where, you have the patient that appears to have a lot of remaining healthy parenchyma, but they have very low, GFR, for example. And what's going on there, right? Is it microscopic cysts? Is it fibrosis? Is it other mechanisms, poor perfusion? So I mean, if I could answer it, I would just say, I think we can provide a lot more information to the clinicians that can help them, provide a more personalized decision and not make it so sort of subjective and qualitative. And that's what I'm hoping we can drive after. But I don't know that there's, necessarily one answer. I just think there's a lot of missing information currently that we're not providing from an imaging standpoint.
I have a question for Terry and for York together. So Terry, during your talk, you mentioned that the mutation in the somatic allele is actually what drives the severity of the disease. And yet we focus on the germline mutation, whether it's truncating or non-truncating. Is there any consistency across a human kidney as to what the somatic mutation is, or is it cyst by cyst? And is there a conflict between these two statements, or I'm just not understanding?
I think it's a cumulative effect of both mutations that drive the polycystin level down below a certain threshold that trigger the initiation.
Right, but is there a consistency of the somatic mutation? Because the somatic mutation is cyst by cyst.
Yes.
So
every somatic mutation seems to be different. There are very few that are recurrent. I don't know if Terry have anything to add.
going to say, if you start out with, let's say you start out with a null allele as your germline mutation, you're already down 50% of the protein. So it doesn't take that much more to go down. I mean, if you say the threshold is 30%, although that could also vary, right? So I think those are related, as York said. It's the cumulative effect. So maybe if you start out with a missense change, let's say the activity of the protein is like 60%, it would take a lot more or a lot more loss of function to get down below the threshold. So I see those things as related.
So if you take two people with a null allele and they have different progression, is that related to their collection of somatic mutations? Is there any, you talked about modifier genes, but we never talk about the somatic mutations in a specific way.
I would say, and York, I'll turn over, that
Yeah.
there could be modifiers that affect the rate of second hits, and then there are going to be other modifiers that are going to affect maybe how fast the cyst grows. But
Yeah, I think I would concur with that. I think the notion that PKD1 is more severe, at least compared to PKD2. I mean, Peter Harris has some data looking at the average cyst number between the two mutation class, that PKD1 patient on average have more individual cysts, like if you certain threshold, like age adjusted, like compared to PKD2. And the explanation was maybe the gene is bigger target and maybe more mutagenic and more prone to second hit. So presumably they have more second hit in PKD1 patient, and that's why more cysts in the former than the latter. I think what is missing is the other equation is what makes a cyst from like tiny to big, because like we could see a lot, some patients have lots of small cysts, but they're not going to develop kidney failure. It's the one that have not just lots of cysts, but lots of marble sizes or ping pong ball sizes that fill up the whole kidney that make the normal parenchyma disappear. And so I think there's this third hit. I mean, Greg's work suggests that maybe that this third hit, whatever it is, whether people postulate ischemic kidney injury, which I think is very probably rare, but maybe Thomas work on crystal, like as a kind of injury of tissue, potentially could provide some more subtle and hidden type of injury that we don't appreciate.
Have a okay. Have a question, follow up to a question from Ron, also for Terry and York. Now, there is at least one study that showed that the cystic cells have hundreds of mutations, not just the second hit. So hundreds of mutations is hundreds of hits, which may involve many other genes. But many of these other genes may also have an effect on the development of the disease, which is kind of very complicated unless we use artificial intelligence for every single cyst, which it's not possible. But yeah.
I was just going to say that in the last study by Hannah Rennert, the one that I presented and I didn't get into it, she actually found a very low frequency of hits in other genes compared with just the random tissue. So I know, but I think I don't know. I viewed her study as being the most rigorous because it used whole genome sequencing. It was like state-of-the-art methods. But go ahead, Greg. You obviously want to jump in. Yeah, go ahead.
I think it's, so I think that's right. I mean, they did not find any in that second study. They did not find any consistent second gene that had a high rate of. There were, I mean, there's certainly any somatic tissue. There's a lot of somatic mutation that goes on all the time in genes. I mean, at a low level, but and can some of them probably affect a ciliary pathway or some other pathway, perhaps. But there was no consistent pattern. The only thing that was consistent was a very high rate of mutation in the PKD gene,
Well,
one or two.
The reason I was asking that is because I'm thinking about, there is a lot of interest now about oxidative stress going on and playing a role in polycystic kidney disease. So if there is a very highly oxidative milieu in the cell, you would expect that you may have a lot of mutations because of that, and that this could be playing a role. But that's
And yet, which is curious though, is you don't see a really high rate of actual cancer, progression to cancer, right? Associated with other high mutability conditions, you normally see cancer as an outcome, and that's not here in general. And so whether it's being cleared because of, again, apoptotic pathways, or because it's just there's not a consistent other pathway that is being knocked off in whatever this somatic mutation stuff is. And the only one that really is likely to be enough to cause it is, a cyst to form, is one that hits that cystic pathway itself directly, I think, it seems.
I guess to make things a bit more
I
complicated.
was just going to say it could be like the same as with any, any one of these cancer genes. You could probably look at BRCA1 or 2, and you look at those cells where you knock out BRCA1 and the somatic hit. I'm sure there's going to be some random mutations in other genes as well, but you're getting a breast cancer. So.
Recently, we have looked at our own data in about 2,000 patients that we had, and from about 600 family, and we have a lot of family that had undergone a gene panel, like 50 genes, just like Peter. So looking at all the possible cystic gene, mostly excluding the ciliary gene, because there are many, and including PKD1, PKD2. So in a group of patients with no mutation detected, we have seen certainly not just like individual, small percentage, I would say 1 or 2% that had 2 different mutations in 2 different cystic disease genes that are not PKD1 or PKD2. So well, this is association. It doesn't prove causality, but I think it just raises that maybe the cystic process can be achieved through different avenue, and that in rare individual, other cystic disease gene can sort of interact, like not just as a modifier, but maybe potentially causal in the phenotype. But most of these few family, the disease are atypical cystic disease, not the diffuse cystic disease as in ADPKD.
The other thing just to point out, I think, is was presented at FASEB, right, was from Peter, the modifier study, right? They have, I don't know how many, there's a couple of thousand people in this study looking at genetic modifiers that might predict more severe PKD. And to date, there really hasn't been a smoking gun identified as a likely primary source, at least on a germline level as a modifier.
I think the GWAS study suggests there's no common modifier, common small effector modifier. I think probably we may see rare higher effect modifier, but they're much less common and not much predictable, making it very difficult to study. So I don't know if there's any other comment. I think we are past our scheduled time, and it's been really an exciting conference, and I've learned a lot. And so I want to thank everybody, and also our virtual audience. Please do fill in the evaluation form to help us to plan for future meeting, and also just make sure that we actually get some feedback. And I guess we'll see you tomorrow morning for those who are interested to take part in the second day. And I just want to mention, it's not too late to sign up for the second day. The second day will also be very interesting. This year's theme is it's called Recovery from the Pandemic and Building Resilience in Mental and Physical Health. And we are very fortunate to have a very outstanding panel to talk to us about mental health and metabolic health. And so I hope that it will be really also an exciting discussion as well. Okay, so thank you very much, everybody.